It's my pleasure to introduce our first speaker this morning. He probably needs little to no introduction to this audience. Dr. Mike Van Amburgh is a professor at Cornell University and a Stephen H. Weiss Presidential Fellow. Dr. Van Amburgh is certainly, he's world renowned for his research and for his teaching and advising at Cornell. So, Dr. Van Amburgh, I'll turn the floor over to you.
Thanks, Clay. Thanks for the invitation to be here. I'm sure this came out of a conversation I had with Clay a few months ago about how we model diets and the fact that we've been caught up in things we call essential, but we seem to want to ignore what we call the non-essential components. And that, to me, is probably misleading as we move into higher and higher levels of production. So I'm going to kind of try to touch on that. I don't know if it'll be satisfactory to everybody. I don't know if it'll be satisfactory to me by the time we're done, but I get to do it again sometime, maybe. All right. So thank you for that. Talk mostly about, I'm actually a little nervous this morning. I don't get nervous to give talks, but for some reason, this one's got me a little, I don't know if it's not enough sleep, whatever. Andrew Lapierre and Andres Ortega, currently a postdoc and a PhD student, contributed to this. So I'll recognize them. So what are we going to do this morning? We're going to talk about essential versus required nutrients. I'm going to talk about implications for both energy and protein requirements. I'm going to have some examples of essential and required nutrients and metabolism, where the interaction might limit productivity, and I'll summarize. I got 32 slides. I should be pretty good on time. Okay. I think it's a real simple thing. You know, when we think about what we're formulating for, most of the time we're thinking about required nutrients, right? And we can do that a couple different ways. I'm going to focus on amino acids a lot today. We could go lots of different directions, but this is easier for me to think about. You know, what is an essential nutrient? It's a substance required by the body for survival, growth, and reproduction that allow for essential functions. That cannot be made endogenously, but can be interconverted to other forms of nutrients. I'm going to come back to that, because that seems to be a real buzz right now, right? We talk about efficiency of use of amino acids, and that, you know, the new NASEM's out. We do that in the CNCPS, and I think we get confused about what that number means. And you know, do you want an optimum output? Do you want a maximum output? Or do you want the highest efficiency? Well, the highest efficiency is probably a deficient animal, right? We all learned that kind of in our sophomore nutrition class, but sometimes when we're out there, we get caught up in these efficiencies. All right, and I think we'll talk about that. Essential amino acids, you know, such as methionine, lysine, histidine, we can go through the rest of them. You know, carbohydrates are essential. Body doesn't make those. I got to figure out this pointer. Minerals, vitamins, things of that nature. Again, I'm going to stick to amino acids this morning. What's a required nutrient? Well, a required nutrient can be made from other metabolites, synthesized or interconverted, right? And an easy example of that is a non-essential amino acid. Is that free? Are those non-essential nutrients free? No. They take energy, they take resources, they take reducing equivalents, they take ATP, they take glucose, they take essential amino acid interconversions, right? And that's all built in when we're trying to formulate for these essential amino acids and their requirements, and we look at these efficiencies. Part of that efficiency is the conversion to non-essential amino acids or any other use that amino acid might have. In my experience with the CNCPS, some of that's really difficult to get to for a lot of different reasons. I'll talk about that. I think we have to start thinking that way. Some of this, I think, can be framed out with the right experiments, and we'll talk a little bit about that. This metabolizable protein thing, right? You know, 10 years ago when we started reframing the essential amino acid requirements as a function of energy in version 7, I had several people say to me, oh, you can throw metabolizable protein out now. My grad students were advocating that. My postdocs were advocating that. And I'm like, I said, democracy doesn't always win here. The answer is no. I let them win a couple times, and it cost us time and energy again because we had to go back and redo something. But, it's the sum of all the essential and non-essential amino acids, right? So when we account for essential amino acids, we assume the non-essentials are met by metabolism as they make up the balance. What we forget is that cattle also consume the non-essential amino acids, right? And some of them are going to get to the small intestine, and some of them are going to be absorbed. And the more of those that are absorbed, the less have to be interconverted from essential amino acids or other substrates, right? So we have this kind of tension going on where those, just because we call them non-essentials doesn't mean they're not required, right? They're required in very high amounts. And I think as we get to higher and higher levels of productivity, all of a sudden those things start to show up, and we don't know how to deal with it. I'll give you a couple examples. So if we think about it, non-essential amino acids generally make up between 46 and 53% of the total amino acid intake, depends on the protein. So they're eating a lot, right? We don't think about protecting them to get into the small intestine, but if you use any rumen-protected protein or any processed soy or canola or whatever, it's not just supplying the essentials, it's also supplying the non-essentials, right? You can see kind of where this is headed, right? So what's one of the next tricks in our modeling effort? Well, we're going to start to frame out the non-essentials. And I'll give you a couple reasons why maybe that should be done. Thus, the intake of non-essential rumen escape will provide MP as non-essentials, just like essentials, right? And, of course, microbial proteins also comprised of both essential and non-essential amino acids. So our problem, we've been a little myopic about this because we even though we analyze for them, we get all the 20 amino acids out of a mass spec or an HPLC, whatever technique you're using, GC, doesn't matter. They're there, we just don't do anything with them. Am I not standing in the right spot here? Should I back up a little bit? Okay. I can see the two of you over there. Okay, I'll stand closer to here. Okay. All right. There's beer in this afterwards, right? Or whiskey, even better. So we talk about non-essential amino acids, right? So I'm going to get into some of the pathways here. It can be made by various pathways using essential amino acids or other substrates. We have to remember that synthesis is energy intensive, right? And this is where, as we look at cows that we are starting to make work harder, not make work harder, they're working harder because we're figuring out how to give them the right nutrients, right? How many of you see milk fats now at 4.5, 4.4, 4.5? Yeah, exactly. That takes a hell of a lot of reducing equivalents. We sure don't want to be spending them on conversion of essentials to non-essentials, right? Maybe a little bit, but we got to be careful, right? Where's the upper limit? So can possibly be limiting under conditions of high demand. Implies, in this, I think this is a big one. It implies a reduced efficiency of use of essential amino acids if converted to non-essentials, as they're not directly used for protein metabolism, right? Because when I mean protein metabolism, they're making a product, but sometimes they go different directions to do different things in cellular structures. Okay. We can follow you. I can stand here. My brain doesn't work as well when I'm standing still, but I'll try. So we get into this concept of metabolic flexibility in the mammary gland and nitrogen use efficiency, right? And there's a lot on this slide. We talk about how adaptable the organ is, and there's lots of sources of nutrients, and the cow can manipulate blood flow. I'm going to come back to the blood flow thing a little bit with one example based on requirements or recognition of varying nutrient supply. The uptake to output of amino acids in the gland is not uniform across the amino acids. I'm going to talk about that based on, MEFM's 1, 2, and 3 groupings of amino acids, right? And we've got lots of different signaling pathways that will play into how the gland responds to nutrients. So our general understanding is an optimal supply of amino acids with improved energy status will maximize anabolic output, right? But notice I use the word optimum, not maximum supply, because I think there are optimums per unit of energy, right? That's just the way we're thinking about it. But there's tweaks around that. So if we look at the mammary adaptability and we think about where things are coming from, here's MEFM's categories, and this modified by Helene LaPierre. So we've got group 1, group 2, and group 3. Group 1, histidine, phenylalanine, methionine, tyrosine, and tryptophan. Efficiency. So we look at efficiency of use, amino acid nitrogen uptake per amino acid nitrogen in the milk. For these guys, it's all 1. If we look at isoleucine, leucine, valine, lysine, arginine, and threonine, all of a sudden it's greater than 1.15, right? So the uptake is pretty high. If we look at the group 3s down here, which are mostly non-essentials, cysteine, we got to flip back and forth between methionine. Proline, I'm going to come back to proline, it's less than 1. Okay? So when we think about extraction of branched chains across the lactation, there's a lot of cellular maintenance and anabolic responses related to the branched chains, especially for casein. I don't know how that happened. And then I lost my pointer. Hello. Oh, there we go. I'll get it. There we go. So, and lysine undergoes obligate catabolism in the mammary gland. So one of Helene LaPierre and I used to have these crazy conversations, and she'd look at me and go, Mike, why does the mammary gland take up all the lysine it sees? And I would just look back at her and say, because it must have a use for it. She goes, that doesn't make any sense. It doesn't come out in the milk. I said, it doesn't come out directly in the milk. That doesn't mean the mammary gland doesn't have a use for it, right? And we confuse that, right? We think, well, if it's an essential amino acid and it's a limiting amino acid, why is it taking it all up? Well, it must have other uses. How many of you know that lysine is involved in fatty acid synthesis? Yeah, those of you that have studied those things. Yeah. So if you're short on lysine, what's going to happen to fatty acid synthesis? It decreases, right? How's that? So because we're trying to figure out how we're getting cows to 4.8 and 4.9 milk fat now. And the only thing we can come up with is we finally gave the mammary gland enough lysine to let it happen. And E. Beauclair would argue with me, that can't be true. Well, I don't know. The cows are telling us it might be true. All right, arginine's taken up in drastic excess, right? 2.5 times. I'm going to come back to that. Catabolism products include proline, ornithine, and urea. Proline is high in milk casein, right? And we can look at, input and output relationships here, and you can see, if you come over to this one, you can see this group 1, group 2, group 3. You can see input and output for the group 1, input, output for the groups 2, input, output for the group 3, right? And you can see subtle shifts in there that say, hey, you know what? This isn't uniform, and they're gonna interconvert a bunch of those amino acids. And you can do this kind of thing here, right? And talk about non-essentials. You can see there's, isoleucine, leucine, lysine, phenylalanine, and tyronine can go into the acetyl-CoA, and off to CO2, can go lots of different directions. Threonine can come in. You've got ammonia coming off. Notice that choline's over here. I'm going to come back to choline at the end of my talk. Tryptophan here can go in to acetylcholine, pyruvate. Here's some other amino acids going into oxaloacetate. The less of these intermediates that we have, the greater the pressure for these things to run that way, which means we're going to use up more of our essential amino acids to fill those pathways, right? Because it's an obligate function. But if we had some other non-essentials in there and some other intermediates coming from other places, then that wouldn't have to happen, right? We get into the branched chain amino acids, and we get into alpha ketoglutarate, the glutamate-glutamine cycle. And down here, you can see the arginine-proline, cycle. So there's all these interactions. I think this is important because it's not a free ride, right? If we start to throw in where we can do the cysteine, cystathionine, choline, get into those interactions. It doesn't really matter. You've got methionine in here, homocysteine, hydroxyproline. In each one of these interactions, you're using reducing equivalents, you're using ATP, branch chain to branch chain keto acids. We're using energy to do all of these transformations. So it ain't free. And as we get cows to higher and higher levels of productivity, all of a sudden we probably need to start considering those. And we hit one of those walls, we think, in an experiment that I'll share with you. Many of you saw last year. All right, if we just look at arginine, this is from a Morris et al. You get diet arginine, you get protein turnover, you get endogenous synthesis. Where's it go, right? Well, in the arginine uptake, again, 2.45 to 1. The range is 0.88 to 4.18, right? So arginine we consider a semi-essential amino acid because it can be produced in the cow. Yet if you look at what it does, it has a lot of roles. Blood flow, proline, polyamines, cell stability, glutamate. I don't think that's really a semi-essential amino acid, right? Just the fact that she can make a little bit of it doesn't mean that it might not be required at some point. Well, it is required, right? The question is does she ever run out of it, and how do we know? Trying to figure that out is the trick, right? So we did this study. This was what we did. I showed this last year. I'm going to come back to it because there's a story here, right? And I think it helps flesh this out, right? So we did this Monensin titration. These cows were making, when we put them on study, sorry, you can't see everything there. They were 90 to 92 pounds of milk, 3.9 protein, 3.9 fat, and 3.1 true protein, right? This wasn't an amino acid study, but we used version 7 thinking to build these diets, okay? There's the composition. I'm not going to spend time on that, but we're trying to be transparent about how we build these diets just so everybody knows what we're doing. Heavy, fatty acid blends, some sugar, getting the amino acids up there, a little bit of rumen stimulation, 54% forage, dry ground corn, a pretty good corn silage and average haylage. Sugar, right, we're pushing that up. Methionine up at 1.19 grams per mcal of Me, and then 2.7 times that for the lysine. So we're pushing quite a few grams, and we're trying to blend up the fatty acids because there's some metabolism there that we think is important. Here's the important part of this, right? So when we start looking at these cows, we're cows 190 days in milk roughly. They're consuming about 26, almost 27 kilograms of dry matter intake. They're making about 39 to 40 kilograms of milk, not quite 40 there. Our milk fats are 4.6 to 4.7, right? Is that pretty good? Yeah. And we took them from 3.9 in mid-lactation. So this is where I think lysine is really important for milk fat synthesis. But you got to have the methionine there too, right? You can't do one without the other. And lo and behold, these girls did what? They made a decent amount of protein, right? We had good fermentation. We must have had a lot of propionate. We had good glucose production. So we're at 3.35, almost 3.4, right? While we're up at close to 4.6, 4.7. Keep going. Energy corrected milk running, in the high 40s, right? Those cows performing pretty well? Yeah, those are pretty good cows. Dave Barbano, here I'll come back to this story. Dave Barbano is part of this. He's calling me up saying, Mike, did you buy jerseys at the research farm? No, I was laughing at him. I said, he goes, why are you laughing at me? I said, why are you asking me that question? He goes, because you got cows that are performing. I've never seen Holsteins perform that way. That can't be true, right? Well, they're really Holsteins, Dave. And he said that we don't have any Holstein data that looks like that, right? And what was really blowing him away were the de novos and the mixed, right? So we're having this conversation, right? And then, I didn't understand this until I went and pulled one of his papers. And here's where his data stops, and here's where our Holsteins are at. here's the jerseys, right? So these cows are performing like the best jerseys. Okay. So this is cool. So Dave thinks this is awesome, right? So Dave's like, hey, this is really great. We got to figure out how to do this some more. Can you repeat this? And then one day he walks over and he goes, I don't like your study. I put my cheesemaker hat on and I don't like your study. I'm looking at him like, it's our study. Excuse me. He goes, not today. It's your study right now. Because I'm a cheesemaker and I don't want to buy your milk. Well, what the hell's wrong with the milk? He says, if I have to make cheddar from that milk, I have to skim it now. Why is that? He says, I can't make legal cheddar with that milk because you got too much fat and not enough protein. I said, what do you mean not enough protein? I said, what does it need to be? He goes, well, that level of fat, you got to be at 3.6 protein. And before I could say what I thought it was, he goes, I think you're short on non-essential amino acids. And I started laughing. He goes, why are you laughing at me? I said, because we just had the conversation yesterday in my lab group. We think we're short on non-essential amino acids. And almost simultaneously, we looked at each other and said, proline. And then we both started laughing, right? Why would we say proline? Why the hell would we say proline? Yeah, it's casein. If there's enough energy there, they could be short on proline because you need to make the three-dimensional structure. Guess what you need to make the three-dimensional structure of casein? You need one hell of a lot of proline. Okay, so let's go there. So if you look at the amino acid composition of casein and bacterial protein, I threw the bacteria on here. Sorry, I didn't have time to do it on the same basis. I just didn't have enough time. But if you look at, I got them highlighted here, is isoleucine, leucine, and valine. These are the amino acids. I'm going backwards here. These are the amino acids. I get my fingers on the buttons in the highest concentration in casein. And look over here. Other than glutamine, 110 grams per kilogram. That's more than everything else. So when you put them all together, if you're making casein, that's 208 grams of per kilogram for branched chains and basically 111 grams of proline. So if you run out of proline, what can't you do? You can't make casein. Now, we don't know this as a fact, but it was interesting that the milk chemist and the nutritional physiologist did what? We came to the same conclusion from two very different directions, right? So all of a sudden we're going, oh, okay, that's interesting. We probably need to focus on this, right? And if you look at proline production in the mammary gland, there is a pathway for this, but notice, alpha ketoglutarate and NADPH is coming from the Krebs cycle, so you need reducing equivalents and you need arginine, right? So there's that arginine thing as a semi-essential amino acid, but it's still implying it's what? It's semi-essential, but it's required, right? And this is one of those things where we muddy the waters by just saying, well, it's semi-essential. I don't know if we really have to worry about it because she's just going to make it. I'm not sure. If we have Holsteins making 4.8 fat and 3.3 or 3.4 protein, maybe she can't make enough, right? Because she's already used up all of her reducing equivalents to do what? Make the fat. The fat was probably easier, actually. And this is another thing because you get into the energy requirements to make protein versus deposit the fat, and they're two different things. So anyhow, so we've got that. We did a study. This is I didn't forget about this. I just didn't really I never had a chance to actually pull it out of the thesis. We did an abomasal infusion study years ago of histidine and proline. And this is CNCPS version 6.1. So this, by today's standards, this thing's really broken. So just bear with me as I go through it, because I would like to take these diets and rerun them to see where we are actually at on a few of these nutrients, right? So we've got this, we're trying to do our best at the time. So we're at 6.1. We got a 635 kilogram cow consuming about 22.6 kilograms of dry matter, producing about 40 kilograms of milk. at 365 fat and 301 protein. We're going to infuse 10 grams of histidine or 20 grams of proline or the same amount of both, right? My desire to want to, the histidine thing was interesting because everybody was talking about histidine at the time as being third limiting, right? So I thought, well, I'm going to throw that in there. My real interest, we just did that because we could. My real interest in this was getting at the proline thing, and this is quite a few years ago now. So we're predicting, right, a negative balance for arginine. So what I'm thinking is I could give the cows arginine, or I could just give them proline to reduce the pressure on conversion, which should take that towards a positive side, right? I'm just playing off the pathway. And then the histidine is the histidine. We know Andrew and I looked at these numbers today, and the histidine requirement's way, way low. So that's okay. We fixed that. So we think we have. Okay, so when you look at the cows, this was a really interesting outcome. They're about 26 kilos, 25. They drop a little bit on the proline. So here's the control: histidine plus proline, and the proline. Feed efficiency, fat-corrected milk, 195, 192, 195, 2.1. Okay, so we popped a little bit, but some of that's the intake. Milk, no significant difference. Fat-corrected milk, no significant difference, but we tended to go up. We're at 52, 51, 8, 50, 49, 52, 4. Fat, 1871, 1806, 1736, 1929, right? On a proline infusion, those girls popped, 50, depends on which way you want to look at it, but you're looking at 50 to 100 grams, not quite 100 grams of fat. Lactose stayed pretty steady. Protein, no big effect, right? But we did see this big increase in milk fat. And again, with the way our study, with the power of it, we couldn't quite get significance there. There is a large increase in lactose. I'm not sure what that means. A difference in PUNs or NPN, right? So we see some subtle effects here. The feed efficiency thing, the fat is the one that really intrigues me, right? Because 1871 to 1804, these are different if you do a mean separation. 1736, that's definitely significant. And the same thing here. So if you do mean separation, these guys are all a little bit different. And the best performance is on the proline infusion, and that's on the fat, not on the protein. That's the other thing I think we have to get out of our heads is if amino acids are not directly related to milk protein. They're related to protein synthesis, but that protein synthesis could be for fat, could be for lactose, could be for milk protein, right? So we have to separate those out. This was the interesting thing. We did blood flow in this study. Here's a control. There's the histidine. There's the histidine plus proline. There's the proline, right? So the cows, and I know some of the Wisconsin data says, well, if you delete histidine, blood flow goes up to respond to it. I don't know if that's what this is, or the cows are just responding to the proline. But you see a nice increase in blood flow here, right? So in other words, the gland is seeing something and responding to it, right? Back to how they sense some of these nutrients, right? So again, proline could be important. Don't have conclusive evidence. We think in our study in that study I just showed you, it's one of the things that may be inhibiting, right? If we look at some recent infusion studies, what's my time here? So we're good. Haha. Well, that's unusual for me. So if we look at the infusions, Metcalf's in the other room. I should drag him over here. If we look at the infusions, he did jugular infusions. There's the essential amino acids. There's the non-essential amino acids. 4 cows, mid-lactation, 4 days of saline, bless you, followed by 5 days of mix. Diet around 87% of the protein requirements and 104 of the ME. Control diet, 104 on the MP and 106 on the ME. MP requirements within treatments, 120 and 108. 120 on the total and 108 on the essentials, right? So when we look at productivity, some of these are a little bit mixed when you look at total amino acids or the essential amino acids. So what you're looking at here is the addition of the non-essentials. And, when you look at fat, and this has been seen a couple times, control versus infuse. The fat actually went down here on his infusions, not on the essentials. Protein increased from the control, right? So both of them gave increases about the same range, a little significance there. A little difference in lactose here. It went the other way. When you look at fat synthesis, you don't see quite as much. Protein, nice push in protein. Same thing there, right? How much of that is it? Oops. How much of that is essential versus non-essential? Looks like a lot of it is essential, right? So in mid-lactation cows, and I think this is one of those things, you're going to see this in a minute, in a mid-lactation cow, is she stressed for nutrients? No. So would I expect to see big changes here? What I wish these guys would do when they do these studies is please weigh the cow. Because my guess is she's doing what? Like all mammals. Yeah, she's putting it on her back. She's not going to waste it. She's not going to make more heat. And I think that's a real big miss in all of this work, is that we're not looking at where she's secreting these nutrients, right? So I think that's something we have to, if we're going to do anything, start weighing the cows. If you go down here, if you look at the essentials, you see a nice jump here, but not so much on the total amino acids. I always find these responses confusing when you just do essential amino acids or you do all amino acids and you don't see the same response. That seems counterintuitive to me, that you're giving it the same amount of essential amino acids, so why adding the non-essentials doesn't seem to take it away, that unless you're just saturating something, right? Here's another one of those studies. These cows are 61 days in milk. They used a casein profile and a 3-to-1 lysine to methionine, and here's the amount of infusion on the essentials and the amount infused per day on the non-essentials. And, again, 8 cows, 61 days, abomacil infusion, 4 treatments, control, essential, non-essential totals, 72% of MP, and they're using 100% of NEL. All right, when we pull this up, sorry, these are a little bit light here. This is interesting. These cows are not eating a lot. They're at 15 to 16 kilograms of dry matter intake, right? So my first, again, but this is one of those studies that we look at. Is there a huge energy demand on these cows? No, right? So I don't want to discount it completely, but it's one of those where I say, these are not high producing cows, right? I got cows that are at 26 to 28 and 29 and 30 kilos now, right? So moving towards twice that, and they're twice as much milk. So I look at this study and I go, this is interesting, but I'm not sure if we're going to learn anything from it, but I'm going to show it to you because this is what's out there, right? You do control versus essential, and you see about 3 kilograms of milk. You do non-essentials, you don't see anything. You do total, and you see 37.9. So you pick up about a kilo from essential to adding the non-essentials on fat from 976. So not 60 grams. Nothing much there when you look at totals. Crude protein, nice jump on the essentials. Not much with the non-essentials. Total, it comes back up. But again, not a lot of synergy here. Lactose, the same thing, right? Fats, these cows are milk fat depressed, 2.9, 2.8, 2.9, 2.6. They're actually inverted. So again, I think they have some probably not great rumen fermentation. There's just a lot of things going on there, right? So I'm not sure this is, again, this is out there. I felt like I needed to show it to you, but we're kind of going, I don't really think that this is probably the right model. Here's the right model. So this is a study out of, I think, Netherlands, Palau, 2021, 9 Holstein cows, calving to 50 days in milk, right? So we're hitting them up, early lactation, non-essential amino acid infusions in fresh cows, 2 treatments, total amino acids or essential amino acids using a casein profile, abomacil infusions, right? Now, these girls, on the other hand, told us what? Yeah, I need those non-essentials. I really need those non-essentials, okay? Early lactation, just initiated lactation, huge demand. So this is the other thing we got to learn. If we're going to study this, where do we have to do it? Early lactation when there's a high demand, right? When the resources are going to be limited. And I think that's the key here is we've got to figure out how to upgrade our model a little bit and be working with cows that truly have a high metabolic demand. We can't do this we can't do it in an easy mid-lactation cow because everybody else is done with them, right? That's not, we gotta kind of get our way in. I always got to work through McFadden and Overton and all those guys and Jess and Sabine who are always doing these fresh cow things. I said, I need to get some of these cows because I need to be able to test this stuff too. Right, but notice what happened, right? Here's the essentials. Here's the total amino acids. You got 9 kilograms of milk. You got 18 to 20 pounds of milk. Holy crap, right? That's a big deal. So can we get rid of metabolizable protein? Absolutely not. This is the best example I have of that. When somebody says, oh, in Amburgh, we can just balance on essential amino acids. We're not going to do that, all right? So, and just to finish up here, one more metabolism slide. I'm not going to spend a lot of time on this. This was from Rodrigo Milano's PhD when he was, or master's actually, when he was working on B vitamins. I did this simply to come down here and introduce this. I'm down in some 1-carbon metabolism here, so methyl donors. And, I know I'm doing a Balchem talk, but I still think this is real and we should be framing this out, right? So choline, is choline a required or essential? Is it required or essential? Is it non-essential? What do you guys think? Come on, somebody give me an answer. You're not going to offend Clay. He's not going to kick you out if you say no. Well, I think it's required. It's definitely essential in my book, right? And because it is essential, what should we be doing in our nutrition models? We should be framing out a requirement. We should be doing that just like everything else, right? It's in 1-carbon metabolism. It makes the efficiency of use of methionine better, right? It's doing all sorts of things that we need to pay attention to. And there's data, right? It'd be one thing if there weren't data, right? But there's data. So I've only got one slide on this, but if you look at the meta-analysis from Arshad in 2020, and you look at, feeding choline and dry matter intake postpartum, you see a reasonable increase. If you look at milk yield, energy-corrected milk yield, and feeding choline, there's a response, right? To me, as a modeler, I look at that and go, I need to figure out how to put some sort of a requirement in there because I think it makes a cow more efficient because she's not spending time using other methyl donors, right? If we don't give her choline, what's she got to do? She's got to find some other methyl donor that maybe is less efficient, right? But if we take it away, we make her energetic efficiency better. If we give it to her, we make her energetic efficiency better, right? And to me, this is all about energy efficiency, right? So put it in there, 1.1 pounds of dry matter intake, 4.8 pounds of energy-corrected milk. Fairly uniform response to me, and this is the conversation I had with Clay a month ago or whenever we had breakfast. I'm comfortable if we can get the right kind of data starting to figure out how to frame this because the data are coming. We got a lot of data now that all say what? Cows are going a certain direction when we put that in the diet. We just, how many grams, when, what does it play off of? Does it interact with methionine? Does it change the methionine requirement? Does it increase the methionine requirement? I don't know. All of those, that's where, Heather, maybe you can help us figure out how to do that, right? And frame it out in a way that we can make a better prediction. Okay, just to summarize, the data would suggest that high-producing cattle have requirements for nutrients that are deemed non-essential. As productivity increases, there are different stages of lactation. Nutrient resources become more limiting for all pathways, and this could be energy, amino acids, or something like a methyl donor. Terms like metabolizable protein are going to remain useful as it captures the supply of non-essential amino acids. And we need to consider non-essential nutrients like required nutrients and start to describe the requirements in nutritional models, right? So I think I'm done and I'm on time.
Perfect, Mike. Thank you. So we have plenty of time for questions. So Scott's going to take questions from the audience here in the room, and if anybody online wants to enter some questions, I will ask those. So Scott, you want to get started here?
Great talk, Mike. Question, and maybe it's intuitively obvious, but I got to ask it. When you see lactose changing up or down, does that imply how she is prioritizing the use of glucose and that she is able to send more to the mammary gland, or she has a requirement for it to do something else? Or can we use it for evaluating efficiencies? I'm just curious. I never thought about it before, but you brought out some points there that made me wonder.
Yeah, it's a good question, Bill. So I'd have to go back and frame that particular study a little bit better. I can use one of Locke's studies, and I'm not going to remember all the details right now, but he was doing some of his fatty acid work, and I kept looking at the diet. He had really nice responses to milk fat. He had some reasonable responses on the amino acid side. It's interesting, if you guys start disassembling the literature and really look at what's going on, I can use amino acids to increase milk fat faster than I can increase protein, and I can use fatty acids to increase milk protein faster than I can increase milk fat. Yeah, think about that for a minute, because it all gets into signaling. Well, both requirements and signaling. To that point, Adam, in one of his studies where he's doing a titration, has a very clear increase in lactose, and not everything is following that. So what that tells me is I asked him, I called him up, I said, did you put any amino acids in this diet? And he said, no, I don't think about that. Why would I do that? That's not my hypothesis. I said, because I think if he had, what would the cows have told him? The lactose went up at least 150 grams, right? Which means milk volume should have gone up, and it did go up a little bit. But when you looked at fat and protein, they didn't go with it, which meant they were probably limited in what? Non-essentials, essentials, whatever. They're limited in something, right? So yeah, if we're seeing more lactose and we're not seeing anything else happen, that tells me that things coming apart a little bit and the gland doesn't have enough resources. I don't know which way they're going to go, right? That's the hard part.
Mike, I've got an online question here. Histidine in MP is low relative to histidine in casein. Is this why cows respond to supplemental rumen-protected histidine, or is it because diets based on grasses limit histidine supply post-ruminally?
Yeah, that's an interesting question. So that original histidine limitation came out of the highly digestible grasses in Northern Europe. So there's a twist on this that I was actually associated with. You won't see my name in it, but I helped the grad student do this. There was a grad student at Cornell many years ago, worked with Jim Russell, and they were trying to isolate some bug. So here's where I think some of this comes from and why it's confusing, because there's a rumen effect that we tend to ignore. Jim Russell had some, in a polite way, he had some biases about how he thought about rumen microbes. And they had spent months trying to find this particular bug that just wouldn't show itself, but it was there and it was an unknown. And one day the grad student came over and he kept showing me his data and nothing made sense. I kept telling him, I think this is not a carbohydrate bug. And he came over with one snippet of data and I said, it's there. This is a protein bug. So I went to a local farm, went to Fezziden's, grabbed some fourth cutting alfalfa, took it home, squeezed it, got the juice out of the alfalfa, gave it to him and said, here, go throw this in your broth and see what happens. The bug blew up, right? It was real easy to find this bug. It was a protein bug. Well, the name of the bug now is Allicinella histiformans. What does Allicinella histiformans do in the rumen? Converts histidine to histamine. So one of the problems with those high grass diets is that bug can convert histidine to histamines, which makes it then even more limiting in those diets. So that's part of it. So we need to protect it, right? We don't always see that because we don't always feed diets like that where that bug is going to show itself, I think, as part of this interaction. So does a cow need histidine? Yeah, I think she needs histidine. I can't figure out, because the data seemed to be mixed to me, right? When I got into a conversation with Helene one day, on that study that we did, she goes, you didn't infuse them long enough. I says, well, what do you mean? She goes, well, you got to infuse them for at least 21 days. For histidine? She goes, yeah, because the liver's got to change all of its enzyme pathways. I said, well, if it's a limiting amino acid, why does it have to do that, right? It should kind of happen relatively quickly. So there's some adaptation things here that I'm still not quite sure about, because I've asked everybody who's done a histidine study, how long did it take you to see the response? And nobody's actually looked at the data that way. So I don't know if we got to be chronic with it. So I think there's a requirement in the way I think about it now. If you're feeding, 78 grams of methionine, you should be feeding about 78 grams of histidine. How's that? That's how I'm doing that.
Mike?
Yes, sir.
So for multiparous cows, that makes sense for a growing animal and their use of amino acids for cartilage and growth, etc. So is the model the same for first lactation versus second, third, and fourth lactations in regards to the use of amino acids?
When you say the model, which?
Well, the CNCPS.
Well, you just have to put the growth requirement in there or use target growth so it calculates it. I think the hard part is you have to have a body weight. And you got to know how much the growth is. What's the growth requirement? There's where we fail. We don't know the body weights. We don't know how much they really have to grow. Dan has pretty good body weights on some cows. He's an exception. Yeah, no, the model will calculate this stuff, and we're working on. So we are working on improving that. We've got new body composition data. The NASM's out. They didn't quite go the same direction we are with it, but we shared the data set. Matter of fact, we curated it and gave it to the NASM committee. So, but we're gonna make sure that we've got those calculations a little more robust in there to make sure that we meet those growth requirements. Yeah.
Other questions?
Okay.
Thank you, Mike.